It's my pleasure to introduce Dr. Vishal Midya, who's going to talk to us about other machine learning applications. So we've heard an overview. We've heard about weighted quantile sum, Bayesian kernel machine regression. Now we'll learn about some other applications. Dr. Midya is a biostatistician and environmental epidemiologist at the Icahn School of Medicine at Mount Sinai, and also an assistant professor in the Division of Biostat of the Department of Environmental Medicine and Public Health. So, Dr. Midya, please.
Thank you.
Hello. Let me move this one here. Doesn't work. There.
Perfect.
That's
good.
Okay.
Thank you. So today I'll talk about the other machine learning approaches. Other machine learning approaches and its relation to mixture methods. So the overarching theme of my talk is going to be slightly different. We'll talk about chemical mixtures for sure, but how these chemical mixtures or the analysis can influence precision environmental health, which is a very relatively newer topic. And with this talk, I want to introduce some of the framework that I've been thinking and obviously future research on that. And we'll cover some of the presentations. We'll go to some recent techniques on precision environmental health. So a couple of years ago, I think last year, Dr. Bakareli and et al., they published a paper on precision environmental health and the approaches and how to approaches to prevent human disease. This paper talks about a couple of approaches and a conceptual framework of what precision health means and what you need to do. So the first one here. So I'm going to spend some time on this slide. So it talks about first the mechanisms by which the environmental exposures can cause disease. We need to understand that. Also, we need to understand the basis of inter-individual differences in susceptibility, developmental biomarker for exposure and disease, and obviously risk prediction using multi-omic data. Taken together, we can do or think about a framework with precision environmental health. Today, in this talk, however, I'm going to mostly focus on understanding the basis of inter-individual differences in susceptibility, the methods behind it, and what does it really mean, and how do we incorporate this understanding of inter-individual differences within the framework of precision environmental health. This is another slide from the same paper, which talks about three different phases. One is the no disease, subclinical disease, and the disease. In the no disease phase, precision interventions to reduce exposure and impact, which many of the methods that we talked about, WQS or the BKM, they create this multi-omic individual risk analysis and the risk scores, and that can be used in this particular no disease scenario. But then comes the other cases where you have some subclinical disease and the other disease cases, and you need to identify those particular subgroups or the specific subgroups which are slightly or more likely have a worse outcome or more susceptibility. How do we do that? I mean, we all eventually are exposed to almost all the chemicals, but not all of us are, or not all of us have a similar risk profile. So identifying the risk profile and also understand the interactions between those chemicals and how did this work, right? So that will be the talk about. So I'll talk about one example first, and then I'll come to the technicality. So the first example goes back to my work with microbiome. So here, what you're looking at is metal exposures during pregnancy, mostly second trimester and third trimester. depressive symptoms in late childhood when the children are around 9 to 10 years old, and the effect of this microbe called Akkermansia muciniphila. Akkermansia muciniphila is touted to be a promising therapeutic probiotic, which have worked pretty well against obesity, type 2 diabetes, and cardiovascular diseases, at least in animal models. And some of the preclinical and the clinical studies have found negative correlations between abundance of Akkermansia and the depressive symptoms in human. What we want to do here is try to see, well, we have seen Akkermansia has some effects, probably has some effects in many of the metabolic disorders. How about depressive symptoms in children? That's the first question. And then if they have this, if there is this association between Akkermansia and depressive symptoms, can it also lessen? Is it true for, is it true for all the individuals, or is it true for a particular subgroup? So, however, can you identify a special subgroup with worsened susceptibility, or the effects are uniform among all? Or third question is, can Akkermansia modify this association? There should be a gap here, a typo. So two questions we want to ask here is the effect of the metals. Is it uniform on all the individuals, or there is a certain subgroup with worsened susceptibility? Or and how do you identify that first? And is this true for everyone? So we'll go back to this cohort called Progress, cohort programming research in obesity, growth, environment, and social stresses. It's based in Mexico City. It was started around 2007, enrolled pregnant women and children being followed till now. And we collected different biomarkers from teeth, blood, and urine. We collected measurements of metals and also some stool sample. So the first thing that we usually do is, well, we look at the individual associations, right? So at second trimester and third trimester, we wanted to see, well, we have around 13 or I forgot, almost 11 metal exposures, and we want to see, well, if they are associated with the outcome. Well, we can look at these plots here. There are some associations, but then it's tricky to say, like, how strong the associations are. I mean, if you adjust for the multiple comparison error, then nothing really shows up anymore. But that's for the overall population, right? But individually, it is kind of difficult to see from here, like, which particular metal affects more. These associations are overall because it's all the whole population, but both for the second trimester and also for the third trimester. However, if you look at the association between Akkermansia muciniphila and the childhood depressive symptom, there is a negative association there. So the question now comes in, well, it seems like the metals individually, or if you do a mixture also, it's not as strongly associated with the depressive symptoms overall. However, Akkermansia is strongly associated with presence of Akkermansia is strongly associated with decreasing the depressive symptoms. So can we find a subgroup of children with a particular pattern of metal exposures who have higher susceptibility of depression, right, from this particular case? Because overall, we don't really see much signals. We found a group with low chromium, low cobalt, and higher zinc. I'll name this group eventually. So kids, so this is a network plot. What we found here is the kids, children who have been exposed to lower level of chromium in second trimester, lower level of cobalt in third trimester, and higher zinc, kids with this particular pattern of exposures, they are more likely to have higher depressive symptoms. This is a little cumbersome, but there are two histograms here. What the histograms are showing is a distribution of the depressive scores for children with the metal clique. Basically means the children with that particular pattern of exposures. And on the right side, this is the distribution of the kids who do not have that pattern of exposures at all. here we're seeing here, the red one basically is showing the kids who have this particular pattern of metal exposures. They're more likely to have lower depressive scores than the other group of kids. But here, the moment you do not have these metal exposures, it seems like Akkermansia municipalia doesn't even work. Like, I mean, obviously there is subtle differences, but overall, the difference almost vanishes. So the point I want to make here is that although metal exposures, we all have certain levels of metal exposures, there are some particular susceptible groups or particular subgroups that we can find or characterize using our exposures. Within that particular group, we can think about possible intervention strategies. In this particular case, it's a small sample size. There is no conclusive evidence, but it can make us think about possible intervention strategies with something like Akkermansia, which is also, again, experimental. But still, this is something you can think about. Now, talking about, now remember, here I presented this idea of three metals. Why three metals and why other two metals, I'll come to that later on. So there's three metals, you can think about almost their as interaction. Now, interaction in statistical or epidemiological term is tricky to understand because you always think interaction as a multiplicative term, and we adjust and put that in our models, right? But interaction as a whole has all different meaning in different fields. For example, in chemistry, for example, interactions, when you are modeling interactions, we have different chemical interactions and the bindings, right? Similar for protein interactions, the proteins, how they different bind. So they are also different interactions. By Aurora et al., they also study a sort of interactions where they talk about metal-metal recurrence interactions, where they are not looking at the level of the exposures, but more about the pattern of the exposures. So that is also another interaction. And then there is toxicological interactions that the other speakers have been talking about, the synergistic, antagonistic, all possible interactions. They only happen when all these metals or all the chemicals are beyond certain thresholds. That's when you see something. And Chris was talking about this something out of nothing. That is kind of the idea also. Individually, you don't see anything much. The moment you put them together above certain thresholds, that's when you see synergistic or possibly antagonistic effect. But as a method, how do you find this, right? Because you don't know a priori what this combination is going to be, what this threshold is going to be. We don't know about those ideas. Just like the Akkermansia examples. So we found these three chemical groups, but we had no idea we will eventually find it, right? How do you find these particular groups? And then again, obviously, look for these interventions. Now I'll go to the methods. So I started using, and I also worked on a couple of these papers. Based on, as Marianthi was speaking, these are based on random forest. Now the idea here is, as she was also mentioning, not using methods directly from other field, because we don't want predictions here. What do you want? The idea, what is happening inside. So we have a forest here, random forest, which basically is a method which can predict very well. But what we have to do here, we have to somehow go inside the forest and understand its mechanism. What we want to do, we want to find a combination of chemicals, could be chemicals, it could be combination of microbes, it could be combination of chemical and microbes that can characterize a certain subgroup of susceptible, highly susceptible, or it could be low susceptible subgroups. And once we find that combination, we can take it out from this forest, and you can use this in any sort of regressions you want. So the idea here is two-staged idea. The first stage, we want to find this combination. Now, there are many different ways to find combinations. There are Bayesian ways to find combinations, and there are many multiple papers. I started using random forest because it's a very natural way which basically creates these branches and bifurcates from them. One example is, let's say I have three different trees. This is like intuitive ideas, right? I have three different trees, and I'm creating multiple different trees here. Let's say in the first tree, On the first node here, I have a chemical exposure called A, which bifurcates based on this threshold 0.2. The next one bifurcates, and there is a chemical exposure B, and the third one bifurcates with the C, and these are the thresholds, right? In G2, I find another branch here with chemicals D, B, and A. Similarly, for nth tree, you can grow multiple trees. You find another branch with B and A. So the point is that it's very natural to bifurcate or create these trees, and the trees are created based on multiple chemicals, talk about chemicals, multiple nodes simultaneously. So the idea of interaction is already incorporated in them very naturally. I'll come about the seed and the other parts here, but intuitively, if we can go inside the forest somehow and pluck these trees like branches one by one, that can give us some idea. But then the next question will be, well, we have so many branches. How do you even like decide which combination to take? Because there are so many combinations, right? That's where the bootstrapping and the other ideas come in. So these are all intuitive ideas. The detailed methods and the mathematics are illustrated in this second paper put here. The next idea is, so this is based on random forest. Now, there are many different ways to create a forest. One of them could be the boosted trees, could be XGBoost, could be multiple different trees. They all have different flavors and dependencies, but for ease of explanation, I'll focus on this is the random forest here. So in this particular example, I come back to the example again. So you see like there are this combination of A and B that co-occurs throughout the trees multiple times. Now imagine I take a data set, do a training and testing and divide it multiple different times. And then in the training data set, I create, let's say 100 trees at each iteration. I go back to the trees and I find like, well, I see that this particular combination of A and B co-occurs multiple times. So although it does not really mean interactions, but there is a co-occurrence. It might also be possible that they co-occur in a different direction. So it might be that higher of A and lower of B co-occurs a lot simultaneously. It might be also possible. This is just an example. So that we find out from here. And we do this multiple times, as Marianthi was talking about. It may be possible that, well, these are machine learning models and they fit really well, even if there is no associations at all or anything to fit, they will probably find some signal. So we have to repeat this whole process many times, the bootstrap, so that we see that these combinations are co-occurring many times, multiple times. And I'll explain like what it means by co-occurring and what it means like finding these combinations or getting a strong hit, right? So this combination of A and B, because it's a combination, it creates a group. I call it a clique. So it is a chemical clique of co-occurrence. So A and B in this particular case, as you can see, A and B co-occurs. So the idea is, well, probably there is some relation between A and B, and we can use it in different frameworks. I'll come to the frameworks later. So this is the idea of chemical cliques. Now, the idea is almost similar to the genetic signatures of genetic, where these cliques are present only in a particular susceptible subgroups. Because these are coming from particular thresholds, you can actually identify subgroups. The moment you have a subgroup, that actually can identify a specific subpopulation. So that's why as a signature in that sense. So I call this a clique signature. It identifies subgroups that may help us understand inter-individual susceptibility. But again, these are machine learning models. We cannot just use it directly. There has to be layers just to make sure that we are not finding any random signals, which is very much possible with machine learning models. So we have to be really careful, like what we call like a signal and what we call not signal. So first is cliques as interactions. So my first example now will be how I incorporate mixture methods and the interactions together in two-stage ways. So we also go back to the here data set. So Chris introduced me the here data set where we get online available data like and many different combinations of metals, exposures, and also disease outcome. So we have been studying this cohort called CHARGE, where it's a cross-sectional study where we have data on kids who have autism spectrum disorder versus controls. And we also have data on pesticides, phthalates, phenols, and trace elements. We wanted to study that whether there is any association between this set of chemicals, pesticides, phthalates, phenols, and the trace elements, and ASD versus controls, ASD being autism spectrum disorder. Because we already have a prior hypothesis in adverse direction, we use the WQS. And as you can see on the left side, we have a very strong positive association here. And this was done on training and testing repeatedly. So the mixture effect of the pesticides, phenols, and the phthalates, they are positively associated with higher risk of ASD, right? It's cross-sectional though. We cannot really say about causality. And we actually found a nice distribution of multiple chemicals. Here there is a chemical called DEP, diethyl phosphate, and there is a chemical called cadmium. The trace metal. Now, DEP had a very high contribution to this mixture index. However, cadmium did not really have. What we wanted to find out, well, does the mixture effect, is there anything, any interaction on top of this mixture effect? So what we did here, we extracted the residual from this mixture effect and used our algorithm. Basically what I told you, right? The RHC. We found this combination called, we found out two-order chemical click of cadmium and diethyl phosphate from the algorithm. So I go to Google and I search like, what does cadmium and diethyl phosphate together mean? There is a compound called chemical diethyl phosphate, and that has been studied for a long time beforehand. So not saying that, well, this compound or the combination you can find in the body or there is a chemical interaction happening, but at least it gives us some way to think differently about interactions without prior knowledge. So we can find some combinations, and obviously we have to go to the biology and see, like, does it even biologically make sense that this compound will fall in our body? Probably even not, but still it gives us some possibility or biological possibility to think about the even mechanism, right? Another example comes from Bayesian quantile sum regression and the use of RHCERF. In this particular example, this also comes from Progress. My colleague Sandra, she was looking at prenatal exposure to a bunch of metabolism disrupting chemical mixtures and how folic acid supplementation eventually affects liver injury in children. So as you can see in this BWQS, which is different from the WQS, here you look at overall mixtures and you combine all the positive and the negative effect together. This is the distribution of the relative weights of the metals and also the air pollutants, pesticide, essential and non-essential metals, and the phthalates, right? What she wanted to look at was, well, is there any particular chemical signature that is there any particular click signature or a particular susceptible group which has a different susceptibility in terms of liver injury on top of these chemical mixtures? And what we found was low cobalt and higher pollutant, and the combinations of interactions between high essential nonmetals, they lead to liver injury. So consistent protective interactions from essential metal cobalt and higher pollutant and also high non-essential metals. So basically, children who had lower levels of cobalt exposure prenatally and higher pollutants, they had way higher susceptibility than the overall population. So this is a combination. Now, why cobalt and how does it interact with possible air pollutants, then obviously we have to think about the biological susceptibility or possibility, like why cobalt has anything to do with liver injury. So we have to obviously think about those biological possibilities, but then it gives us a hypothesis to follow through. Another example comes from Nuria. She was looking at prenatal exposures to endocrine disrupting chemicals and the obesity risk in childhood and adolescence. She also did a mixture analysis for metals, pesticides, PCBs, phenols, parabens, and then on top of the mixture effect, she tried to find any particular combinations. Here also we found a combination of high cadmium and high PBD, and high cadmium and high DDE. So kids who have this particular combination, which is almost 1/4 of the sample size, they had higher susceptibility. And this had been done in training and testing. So with these models like this, if you fit these models in the whole population, you're definitely going to find a signal, because it's a machine learning model and it will predict something. So we have to be really careful to have our training and the testing and possibly a validation data set, so that whatever we find in the training can also be tested and validated in the other samples. And the next examples come from the microbial exposomics. This is my work with my colleague Shona and Jamil. I'm gonna skip this. So here also the idea is that not just the chemicals, but also the gut microbes, they also work in a synergy. So they also form colonies or the groups. The idea here is, again, we don't know a priori what the group is going to be, but we know that there might be a synergistic or antagonistic effect. So we need to find this particular set of combinations of microbes. So the whole idea is finding this group of microbes, or the microbial cliques in this case is gut microbial cliques. So we did a work last year where we wanted to find how prenatal lead exposure is associated with later gut microbial abundances in the childhood, right? So what we found was exposure to lead obviously is or not obviously, it could be detrimental. And in this particular case, what we found is high prenatal blood lead exposure can lead to lower abundances in a clique, a combination of probiotic gut microbial clique, even after 10 years later. But if you look at these associations individually, I mean, you will see like decrease, lower abundances, but it is way more prominent the moment you start forming these cliques or the groups. So as overall, exposure to lead affects strongly on the group than individually. And that's what we wanted to find. What happens to the group combinations? And in many of the cases, the gut microbes or the gut microbes, they actually feed on each other and they have different functions together. So you want to find that with this clique idea. But another point of the clique is they need to be very much interpretable, right? I mean, we can use other models like principal component analysis and also different clustering algorithms to find these combinations, but then we lose interpretability. What we really need here to find this clique, because we want to find this susceptible subgroup so that we can probably intervene and also probably understand biological mechanisms, there need to be interpretability. So that's when this clique method comes in, where we can go to these branches of the trees and then we can pick these branches and look at these combinations. Another example comes from these metal microbial cliques, where we studied prenatal metal exposures and how it affects childhood depression index, metals, depression, and also gut microbial clique. Metals can affect the gut microbes, and then gut microbe has this gut-brain axis where through it can affect the childhood mental health. We wanted to find, well, if there is a synergistic or antagonistic association that is prevalent there, and that might overall affect the depression of our children. So in this particular case, we found a combination of zinc, cobalt again, and there are two microbes, Pranuzzii and fragilis. Combination of this, we found that this particular combination was extremely prevalent in our sample. And then kids who have this particular metal microbial clique, which is basically shown on the right side with the red histogram, they have much higher likelihood of having, not like they have higher score on the log transform T score than the kids who don't have the click. And you can find this on training and the testing data. So we did it multiple times just to see, well, this is even valid for overall data set, or is it basically like a certain subset which you are basically seeing this association. So we have to repeat this repeat this process multiple times throughout different training and testing iterations. But then even then, you actually find this particular combination. And then eventually found, well, why cobalt and zinc? Cobalt is a part of cobalamin, which is a part of vitamin B12. And it seems like vitamin B12 has a strong association with depression index. So it might be that cobalt in prenatally and zinc and this combination of bacteria that might have a synergistic effect, which actually for kids with this particular chemical microbial clique has this particular synergistic effect, which can possibly have a worse outcome on the depression index. And then the another idea of clique is that once you have these particular combinations and the cliques, you can look at the individual associations. You can look at the individual associations of each of the components, right? What does it mean to have high zinc? What does it mean to have high of these probiotics? What does it mean to have low cobalt, for example? So we can actually look at individually what it means and also for each of the combinations to understand how it eventually affects. Now, obviously, we have to control for multiple comparison error and all of the other stuff. But then you see, the moment you have these overall 4 combinations, that's when you have the highest, strongest effect. And they're, again, interpretable, right? The interesting part here is if you look at this combination, if you look at these components just by itself, they're not very correlated by themselves. It's only when they come together, that's when something happens, or like something out of nothing, almost. So if you look at these correlations, they're very low correlated. So these combinations are not just formed based on correlations, rather it's a possible interaction or synergistic effect. Now, I'm saying interaction in a very loose way. You have to obviously allow it with a after controlling for the main effects. But you can actually find these combinations through these tree-based methods. And then I have the chemical click signatures. Chemical click signatures that I have been talking about, this is another example of chemical click signatures. So I went through the microbial signatures, but this is now chemical click signatures. We also did another study where we wanted to see if metal exposure during pregnancy and childhood asthma. And in this particular case, we were looking at metals from urine, and we wanted to see if there is a mixture effect of these metals and asthma risk in children, and whether there is any particular combinations or the susceptible groups who are more susceptible to asthma risks. And the susceptibility can be characterized with these particular metals, right? So we did a mixture. Within the mixture effect was not associated with the asthma risk overall. However, what we did was we found these click combinations of aluminum, zinc, and manganese, and its association with asthma and as well as association with wheezing. So asthma and wheezing, they go hand in hand very much. In this particular plot, on the left side, we are showing 2 different distributions. One distribution is kids with no wheezing, when they have the click, and the right side is the no click group. As you can see that the moment you have the click, the possibility that you have or the prevalence of having higher asthma or any or every goes up, having the click with respect to not having the click. Similarly, on the other side with asthma. So the point is that we can obviously find the mixture effects, but also we can find these subgroups which are more susceptible. And then once you find these susceptible groups, you can always go back and look at the linear associations or nonlinear associations. It's basically finding out these particular groups. That's the most important part. And then we, in the plot C, we plot these associations individually with the clicks, right? So having low aluminum, low zinc, and low manganese, and their association with wheezing, and their association, the same click, and their association with asthma. So the click has not been changed, but then these two particular outcomes, they are strongly associated with this particular with these combinations. And there, if you look at the Spearman correlation between these metal clicks, they are not as strongly correlated, at least the aluminum, zinc. Manganese has some correlation with, in this particular case with zinc, but not as much. Obviously, the click is correlated with all the metals, but individually, they are not correlated. So component-wise, the correlation is very small, but the moment they come together, that's when you see some strong effect. And the last example is based on metal dysregulations. So dysregulation in metal and fetal biodynamics, this is a newer idea where we are not looking at concentrations, rather we are looking at the biodynamics of the metal exposures. So here you see these two plots about lithium exposures in teeth, and we're looking at exposure of lithium in teeth and depression index. On the red side, this is a exposure pattern, which is slightly dysregulated. On the right side, this is a nice regulated dynamic distribution pattern. What we wanted to see, well, these patterns, how they, if you have a regular pattern, is that regular pattern somehow related to depression, higher depression score. So dysregulation in fetal metal dynamics on both sides, if the pattern in the metal exposures is somehow associated with depression or the anxiety index, right? So we looked at this in many different settings with lithium, manganese, and we also have like 17 metals. Individually, there was no strong effect, but the moment we study lithium and manganese simultaneously through the click, that's when you see a strong association. And again, similar kind of plots here. Children with greater consistency in lithium and magnesium, they actually are more likely to have lesser score on the depression index with respect to kids who have higher inconsistency. And you can actually, again, once you have this particular combination, you can actually plot them and see their association estimates. So the overall idea again here is we find these clicks with this particular, with this, we go back to the, here. We find these particular combinations with the method called random forest. It's iterative random forest, but you can also use methods like XGBoost and others. And then once you go to the forest, you basically pluck these combinations or the branches. Once you have the branches, the most co-occurring branches, that's when we find, once the branches are there, For example, here you have the ABC and the different combinations. Once you have the particular combinations, you take this particular combination and then find the effect estimates in the next stage. So that is the whole idea here. There is a nice vignette I have created in GitHub. So you can go there. I have also simulated data sets there, and you can actually use those simulated data set, use that particular, all of these methods, and you can actually even change the code if you want. Because this is a machine learning method, we have to be really careful of how we are implementing it. Because again, there's a good possibility that even in a data set with no possible association, it will probably predict. So there has to be, as Marianthi was saying, there has to be training and the testing, validation, and it has to be done many, many times. So we usually do more than 100 times or 500 times just to find a possible strong association. There is another possibility that we can use is a permutation test, where we can actually permute the exposures and also probably permute the outcome, do this same method and try to see the click that I found here, can I find it there also? That possibility is not, probably won't find it, but if you do find the same thing in the permuted data set, then your click is probably not the correct one. So there are different sensitivity analysis that we have to do just after doing this method. And there are different extensions that can be done from here in future. And yeah, that's it.
Well, thank you, Dr. Midja. Very nice. So let me start off with a question. You didn't say too much about how you would implement this. Is there some useful R packages or packages that you can use?
The R packages are based on, this is my email. The R packages are based on, so there have been codes for the random forest, which is there, and also for the iterative random forest. So the vignette has already incorporated many of these R packages. I'm currently working on creating a R package and incorporating all of them together. But in GitHub, there are multiple pages where I have with examples and written down how to implement them with WQS, BWQS, and G computations. Yeah. So it's in future work.
Great. Okay. We did have a question from someone online here. It reads, random forest methods are known to have many parameter settings. Can you talk about the robustness of the results obtained from the SIRF method? And how sensitive is the method to different parameters of the algorithm, such as number of bootstrap samples, tree depth, choice of node purity measures?
Very good question. So that's when I think the idea of multiple training and testing split comes in, and multiple bootstrap iterations. So usually I do a bootstrap iteration of 200 at each time and repeat this process at least 500 times or 200 times, depending on the data set, size of the data set. The idea is not to create the optimal prediction model. Rather, the idea is to find the perfect combination, right? So usually it is not about the hyperparameter or the tuning as such, but more about going into the forest. So I usually fix my hyperparameter. And once the hyperparameters are fixed, it's more about, am I seeing this signal in multiple different training and testing splits? If I'm seeing it, that's probably is valid. If I don't see that many, many times, then it's probably not valid. So and to answer the question is, the hyperparameter tuning is not probably the main part here, because I'm not trying to create a prediction model, but rather keeping everything at their baseline or the possible what they actually, it's possible like the minimal setting. And then once we fix that hyperparameter, we do the bootstrapping many, many times. Yeah.
Okay.
But more than 100 times for sure.
There's one follow-up question, or one additional question, part to the question, I should say. Along the same lines, have you investigated how well the identified clicks generalize beyond the data set they were trained on? So I guess maybe that would be your
Yeah.
testing data set.
Right. So it will be nice to have multiple cohorts. And once you have multiple cohorts, obviously, if you find a certain click in one data set, you can actually go and try if the association is there in the other data set as well. That another possibility is that I have been working a lot with the depression index or the mental health outcomes, but then there are multiple scales there. So if you start your model with a particular index, you can always find the click and try it with a different index. So the outcome changes, although there might be some correlation between the outcomes. However, if the click is valid for one outcome and the outcome is conceptually related to the other outcome, probably we'll see association with the other outcome as well. So using conceptually similar outcome can also help. Sometimes it's not always possible to have similar cohort at the similar data structure. It is very difficult. So that we have to keep in mind. But yeah, training and testing within the cohort sometimes helps. But again, there is always the limitation of geographical location.
Very good. Well, seeing no other questions, I think we can thank you and end this part early. So thank you again.